from Professor Menachem Moshelion from the Hebrew University in Jerusalem, talking about guidelines for designing drought experiments in controlled conditions.
Thank you. Actually, I'm not going to present today my work, but more of editorial we are just releasing from the JXB. The editorial name is Guidelines for Designing and Interpreting Drought Experiment Control Conditions. And we wrote it together, me and my four colleagues, Professor Carl Joseph Dietz, Professor Ian Dude, Professor Bertan Muller, and John Lund, which is also the editor-in-chief. So why to write this kind of guidelines? Well, if you look on the acceptance of papers to our journal, you can see that we get abiotic stress double than the biotic stress. Okay, this is what submitted, sorry, not acceptance, but the acceptance rate of the biotic papers are 50% higher. Okay, that means that something in the process make the abiotic stress being rejected more. If they're rejected, they're probably going elsewhere, right, to some other journals. So if we look on the general publishing, you can see that in the global publishing, you see much more abiotic stress published than biotic stress published, right? So in gray, you can see the abiotic papers, and in the black, the biotic papers. But if you look how much patent translate from it, the number are equal. And if you actually going and check how much of this became product, only 7% of the abiotic related germplasm and so on became real product. And we all know there is a problem, right? If you look on this biodata, you can see that under drought, the majority of loss come from abiotic factors like drought and heat, and very small portion been lost due to biotic stress. So in research, we have some problem with this abiotic stress. And when we try to analyze what is the main cause for the experience, at least from what we get in the paper, we came up with these four problems that we think that everyone that doing abiotic stress should address before even start to designing its experiment. So these problems are basically related to the complexity of the plant-water relations, right? We all know this kind of figure that, you know, plant, water, sun, but I like to think about this water balance more like this bicycle balance. You cannot understand it just by looking at it, right? So this plant-water balance is a very complex thing, and I want to give a very short introduction. Plants lose water via the stomata. At the same time, they gain CO2. The problem or the issue here, the ratio, is about 500 to 1. This is average. So plants lose 500 molecules of water to gain 1 CO2 molecule. Okay, this is the basic water balance. And when we look at it and we think about plant-water balance regulation, this is what we should keep in mind. So if you look on the wild type probably adapted to his environment using the sources he has, and he reached to some kind of optimal behavior in the environment. But when we look on breeding pushed the plant very strong to produce more yield. And many times we find that these bred crops has more stomata, they gain more CO2, but also lose more water. So if we look on, for example, this graph, you see these are bred wheat line. You see as time passes, more and more yield being produced. Very, very logic, very good. But if you take the same lines and you check their transpiration or stomatal conductance, you find even a deeper slope. So the modern line, these, produce more, but also lose much more water, okay? So if I need to sum up this, breeding, the yellow arrow here, bring more yield, this is clear, but also usually reduce the water use efficiency and make the plant to take more risk. What do I mean by risk? Because they open stomata longer, they lose more water, and they are under more drought risks, okay? And these things must be taken in account also when even starting to design the experiment. So in our guidelines, we suggested what we call the 4D approach or checklist before you even start to think about your experiment. Define, develop, design, and devise. I will just go through this one by one. When I say define, first, think about what is your research question problem, not just in a wishful thinking, I want to save the world, but also maybe very practical, okay? For example, if a classical question said, can we improve the yield production under drought stress, right? This is like, I would say, the question for most of the papers. Try not to use like, we're going to develop plant resistant or plant tolerance. This is too general and have very little information inside, okay? Try to be more specific, like, can we, or try to put more quantitative, like, can we improve photosynthesis or the root lengths to reach deeper water? Something that could be really measured and explained and not just drought tolerant. When we talk about hypotheses, of course, when you do research, usually you need to have some prediction or hypothesis. Here again, in the terminology, we see very fantastic diversity in terminology, like very basic stuff. For example, if we take a plant and we expose it to abiotic stress, drought in particular, if a plant dies, that means it's susceptible. Many papers call it sensitive, but it's just the opposite. If the plant was sensitive to the drought, it probably sensed it first, and then it would activate some escape mechanism or any other defense mechanism to enable it survive the stress. Now, this is very good, but this is not agronomic. Because if a plant just survives the stress and you show us like a dead plant and a live plant, this is fantastic, but you cannot come to the conclusion that this is how you're going to save the world. Because if a plant just survives, now we have to recover, what we call resilience. Resilience, the best definition is the recovery from stress. Many times I see resilience in totally different aspects. Now, even if you recover from the stress, from the ecological point of view, you can say this plant is resistant, right? He resists the stress and he didn't die. But from the agronomic point of view, it's actually meaningless because we can say he has some yield or maybe tolerant the stress, but from the agriculture point of view, this is not a good trait. Sometimes even the opposite. And so on and so forth. Actually resistant, drought resistant, to my opinion, there is no such thing. So if you say drought resistant, I don't know what you mean, because drought resistant means that under drought and non-drought, it should look exactly like the control plant. And I don't know how this can be. So check your terminology. It just helps, to compare papers and really understand what you mean. Design. This may be the key aspect of any good experiment, and of course for abiotic stress experiment. First of all, we need to think what are the response variable and what are the controls. Like any other experiment, we must remember that when we do drought experiment, it's not just the water. Plant responds to many independent variables from the atmosphere, like radiation, of course, but the radiation is usually not steady, right? So it changes, and it affects the temperature, that affects the relative humidity, and these two affect the VPD. VPD, I would say, is as important in drought as the drought. Because, for example, if VPD is 0, no demand, the drought is almost meaningless. The plant will hardly transpire water. So try to address this aspect, even if they are not really important, at least measure them and report them. And of course, the water in the soil and so on, I will get to this better later. Why is it so important to address this? Because the plant responds to this condition. Any one of this radiation, temperature, relative humidity, the plant responds in minutes. And of course, we have stomatal aperture, which lead to transpiration and so on and so forth. So what you have in the end is a very multi reaction and a complex phenotype that usually it's not enough just to measure one thing. So when we ask for the design, first of all, you must remember that each plant in your greenhouse or your chamber probably feel something a bit different. It could be different temperature, a bit. It could be 1 degree or 2 degrees, maybe a bit different of light. Usually there is not too much to do about it, we know, but use the classical randomization, okay? Build an experiment with randomized structure. I know that when you do irrigation, it's more easy to divide 2 blocks to put one pipe to the left and the other pipe to the right. This is fantastically easy, but it's not true because maybe all the plant to the left suffer or sense very different condition. So randomization is very important. Measuring the soil, plant, and atmosphere condition and reporting them, okay? It's not enough to say drought. We need to see what was the water potential or water content in the soil. Then your whatever biometric test that you take for the plant, it could be whatever you want it to be, and also the atmospheric. So try to measure the soil, plant, atmosphere as often as possible, because this is a very dynamic procedure. And also remember that the trait you measure are very important. So how you measure it, what you measure it, and when you measure it is important. So try to report it. Doesn't matter if you use imaging or physiological measure or biochemical measurement. So I want to give you an example about very, very popular measurement, which is phenotyping using RGB. So if you look on this well-irrigated plant and you expose it to drought, you can get 2 phenotypes. Which of these would you choose as a drought better response? Most of the people say the upper one, but this is just isohydric plant. This is a plant that closes tomato very fast. It's not good agronomical trait. While the lower plant closes tomato later, which means is more productive. In this state of the leaf now, it has like 90% photosynthesis. For example, if you look for a worker in the field, right? You come in the end of the day, which one of the worker would you choose to be the next day worker? The one with the clean hand or the black hands? Most of the people will take the black hand, right? Because he work harder. This is the same thing with plant. We want for agronomical use, the plant that opens tomato more, longer, but then they will be more susceptible to drought. So this is the risk I talked about before. I don't say I have a solution what to do, but at least report it and measure it. Know that these changes are there. So maybe this is not a drought tolerant or resistant. This is just drought survival. And as I told you before, we don't always really need that unless we are working on ecology issue or, I don't know, perennial issue just need to survive this season and produce the next season, okay? So let's go to the final thing. Okay, so after I design an experiment, how do I really control everything? How to really measure all these things? So of course, there are all kinds of ways. We know some of them are very expensive, but the most important to remember that you want to compare apples to apples. So if you compare several genotypes and one transpire more than the other, don't compare them based on time because the one that transpire more will be exposed to drought faster, which will cause bias. Let me show you in this small cartoon. Let's say I have this cultivar that transpire a bit more, you see 140 ml, than this cultivar. Now, if I do a dry down, which means I'm just closing the irrigation, the plant that transpire more will suffer faster from drought in the same pot, in the same conditions than the plants that suffer less. So if I compare the transpiration, of course, the plants that transpire more and actually more productive will lose transpiration faster. Another bias is temperature. If you expose your plant to similar temperature, like in this experiment to 35 degrees, it's not enough to measure the chamber temperature. You have to also measure the leaf temperature, okay, and report the delta between the leaf and also report the VPD. It's almost impossible to control the vapor pressure deficit in chamber because when you change the temperature, you also have to change the relative humidity. And this is tough. So if you cannot stable it, measure it. How does this look in real life? If you take tomato, what you see here is the population interrogation line of tomato. If you just expose all of them to drought, this is the differential in behavior you see because of transpiration, only transpiration. These plants are similar in size, only have different transpiration level. So if you just go and measure extract your RNA in day 18, and you do it to all the plants together because you want to keep it very similar, some of the plants will not be exposed to stress at all. Okay? Of course, how to measure it, how to do it. So there are very nice phenotypic machines. I know they're expensive, but today there are protocols, calls like AgroServe that you see here. I even copied the website. There is now a call that you can go to any one of the 63 phenotyping machines in Europe. You just apply to this call. You say, I want to phenotype my plants. And if you write a nice proposal, you're going to get access and fully paid for this machine like the one in Netherlands. You see, this movie doesn't want, but this, in Netherlands, they have this lysimeter machine that each plant can be irrigated and maintain the soil water content as you wish. Everything is automatically done because there is another system in Israel. You can just apply this AgroServe, say, I want to work in this facility, and if your call will accept, you can use these facilities. I'm not going to get to the technical details, but these facilities control each plant by its own transpiration. So it's not just by the sensor. The control is a feedback from the plant transpiration, so you can get a very standardized experiment. This graph that you see here is the same tomato line I showed before, but now look on the soil water content, how homogeneous it is. You remember before it was very widely open, and now due to this machine, you can really compare the same stress both in the soil and in the atmosphere. I'm short of time, so I'm just wanted to, another last thing. When you compare, when you measure plants, try to measure the physiological trait and which is the dependent and, the independent, like the soil water content together, and decide the stress based on the physiological point, which is usually when the stomata is closed. That's it. I finished here. So just remember, next time before you start your experiment, please go to our website, read these guidelines, see that all of these lines are checked. Again, thanks for my co-authors, and thank you.
Thanks a lot. Yeah, since this is the last talk, we may have time for 1 or 2 questions.